Mr. Chairman, honoured guests, members of the Nepal Mountain Medicine Society, I'd like to start by adding my welcome to those of my colleagues and friends to this joint meeting of Xtreme Everest and the Nepal Mountain Medicine Society. It's a great regret to me that I can't be with you in Kathmandu, but I'm afraid the arrival of our third child precludes that. But I wish you, with my colleagues, a very productive and enjoyable meeting. My role today is to introduce to you Caudwell Xtreme Everest, which was this huge research expedition that we were involved with in 2007. And I'd like to start that by showing you a short sequence of video, and it's a video of a climber close to the summit of Everest. And I'd like to show you this video because it really highlights the profound disability of individuals in this kind of environment. And in particular, there's an individual who I will draw your attention to, wearing a blue hat, who we'll see about 25-30 seconds into this sequence. And this individual is a competitive fell runner at sea level. So he's a county standard athlete who does a sport which is common in the UK but probably not so common elsewhere, which is running up and down mountains for pleasure. And I will highlight him as he comes in the video. So what you can see here is, first of all, a climber approaching the Hillary step, attached to the fixed ropes. The images you're seeing are from a head cam attached to the helmet of one of our Sherpa colleagues. We're now above the Hillary step, still on the fixed rope, probably only a couple of hundred metres from the very top of Everest. This is the view over towards Makalu on your left, and in a moment you will see our friend in the blue hat with supplemental oxygen taking the last few steps up to the summit of Mount Everest. This is the summit of Everest, festooned with the prayer flags. We reached the summit of Everest towards the end of the season in 2007. And these are the last few steps up the ascent route from the Nepalese side of the mountain. And in a moment, the first views, or the first clear views over onto the vast, high Tibetan plateau at 3,500–4,000 metres as this climber reaches the summit of Mount Everest. Mount Everest, 8,848 metres, 29,028 feet high, as we all know, the highest point on the surface of the Earth, but also, by strange coincidence really, absolutely at the limit of what humans can tolerate in terms of low oxygen levels. The view you can see here is the view, as I'm sure many of you know, from the Nepalese side of the mountain, and we can see highlighted on the left-hand skyline of Everest the North Ridge. And the North Ridge was attempted a number of times in the 1920s and the 1930s by British teams. British teams had unique access at that time through Tibet to the north side of the mountain. And although they weren't successful, one of the remarkable things about that time was how close they came to the summit of Everest, both with and without supplemental oxygen. In 1950, things changed abruptly. Tibet was invaded by China, and the northern approaches to Everest were abruptly closed, and very shortly thereafter the Nepal south side approaches to Everest opened up. And so in 1950 and 1951 there were early reconnaissance expeditions. In 1952 there was a very serious attempt by the Swiss, Raymond Lambert and Sherpa Tenzing Norgay, reaching very close to the summit of Everest using the southeast ridge route. But unfortunately not quite summiting, and then as you all know, the first ascent of Everest in 1953 was by Sherpa Tenzing Norgay and Edmund Hillary, and they reached the summit on the 29th of May. And as you probably know, the news then reached back to the UK. This was a British expedition on the eve of the coronation of the current Queen. Now, the thing to understand about this ascent, and this is a picture of Tenzing taken by Hillary on the summit, is that both of these climbers used supplemental oxygen. And if you read Hillary's account at the time, he took off his supplemental oxygen for some time and describes feeling rather woozy and uncomfortable. And his view seems to have been that it might not be possible to climb Everest without supplemental oxygen. And that probably accords with the bulk of both medical and physiological opinion at the time. And it took just under 25 years, 3 weeks under 25 years, for that question to be answered. And in May of 1978, Reinhold Messner and Peter Habeler, two German-speaking Italians from the Tyrol, successfully reached the summit of Everest without supplemental oxygen. Now, for most of us, that would have answered the question as to whether Everest could be climbed without supplemental oxygen. Clearly, from a physiological perspective, it did. Intriguingly, from a climbing perspective, it generated some controversy. Because Messner and Habeler, many of us would argue prudently, had some oxygen available to them left on the South Col, the climbers, or some of the climbing community, suggested that this meant that somehow this was an oxygen-assisted ascent. Now, Messner, being, I think, somewhat more cussed than the average cussed climber, decided that he would solve this argument once and for all, and he came back in 1980 and climbed in the summer when nobody else was on the mountain an entirely new route on his own on the north side, and finally put that argument to bed. So my role today is to introduce the project and explain to you why we undertook medical research on Everest, why we took this large number of healthy volunteers to this remote and harsh environment, and secondly tell you a little bit about what we did on Everest by way of introduction for the following speakers who will tell you about much of the science. So why do medical research on Everest? Well, there are two principal reasons. The first is that we felt and still believe that studying humans progressively exposed to hypoxia at altitude was a useful model for understanding what happens to them in critical illness. The second reason is that our understanding of the classical models of what happens in terms of adaptation or acclimatization to altitude was that whilst they were a good description, they really were not an adequate explanation of what happens to humans at altitude. And I'll take those two in turn. It is an almost universal phenomenon in critically ill patients that they experience cellular hypoxia, and in many cases they also suffer from hypoxemia, low blood oxygen levels. Now, the problem about studying critically ill patients and understanding this is that they don't come like this. They're a highly heterogeneous group. They're heterogeneous in terms of the variety of different complaints that bring them to a critical care unit, the variety of different conditions that they had before they developed those conditions, and then all the spectrum of treatments that we institute and their responses to them. And the consequence of all this is that studying the effects of a single variable or changes in a single variable, like for example the level of oxygen in the blood, hypoxia, is very difficult. It's very hard to separate out the causes and consequences of an isolated variable. So we took an alternative approach, and that approach was to study healthy volunteers ascending to altitude, where the principal difference over time was that they were progressively exposed to less and less oxygen, more and more hypoxia. And an added advantage that we had with that approach was that we were able to get baseline data. We could study these volunteers in London and understand what their physiology was normally like, and then compare their physiology at altitude with their normal physiology. And that's a luxury that we rarely have in critically ill patients. We usually meet them when they're already unwell. Now, you might argue, or you might ask, are there not other ways of studying critically ill patients? Now, there clearly are, but one of the problems of critical care research over the last 20, 30, 40 years is that we've used alternative models, we've used animal models, we've used benchtop preparations, we've used so-called in silico studies, modelling using computers, and at the end of all that, we do not have a single specific therapy for a critical illness. So we don't have a specific therapy for severe sepsis and septic shock. We don't have a specific therapy for the acute respiratory distress syndrome, for example. So critical care is a speciality that is supportive. We have many supportive measures that we can apply, but we are unable to treat the specific syndromes of critical illness. So we've taken the approach that the best model to study is humans rather than any other species, and that studying integrative physiology allows us to understand the different relationships between various organ systems and metabolic systems. And having said what I have about patients, we've taken the approach of studying healthy volunteers and altering the isolated variable of their degree of hypoxemia. So is there any validity in this model? Well, I think there is. I think there are parallels between the physiological and pathophysiological processes that we see in critical illness and the processes involved in adaptation and acclimatization to altitude. And there are some intriguing parallels in terms of therapies in critical illness. So what we see in acute injury, and I make a clear distinction here between acute injury, the first few hours, perhaps the first day after an injurious stimulus, which might be trauma, it might be acute sepsis, acute infection, or it might be major surgery. And the classical response to this is a fight or flight response. So it's an increase in adrenergic output, an increase in cardiac output, oxygen delivery. In contrast, in established critical illness, we see something completely different, and that is we see in general a down regulation of activity. So something more akin to a hibernation type response. Now, the same patterns pertain when you look at individuals exposed to hypoxia at altitude. The acute response is very typical fight or flight, but the more sustained response and the responses of acclimatization and adaptation seem to be much more about down regulation, again something akin to hibernation. Intriguingly, if we look at the application of therapies which are designed to increase oxygen delivery in different clinical contexts, in acute injury, it seems that there is benefit from these type of approaches, and that's true in acute infections, so severe sepsis and septic shock, in acute trauma and following major surgery. In contrast, in established critical illness, there have been a number of studies which have clearly demonstrated that this type of approach of increasing oxygen delivery is not of benefit, and in fact in some cases is of harm. And the two graphics you can see in front of you, on the left hand side is an example from major surgery, with the blue and red lines being intervention groups who've had their oxygen delivery increased, and the black line being control, and you can see improved survival in the intervention groups with an increase in oxygen delivery. And on the right hand side, the graphic is of a similar style of study in critically ill patients, and the upper control line is those patients who receive conventional therapy. The lower darker line is those who had their oxygen delivery increased. So a clear contrast. Furthermore, and very intriguingly, there is fascinating evidence from genetic polymorphisms, and this is data from my very good friend and colleague Hugh Montgomery, looking at the ACE gene, or the angiotensin converting enzyme gene. And he looked at different alleles of this gene, notably the I and D polymorphisms. And these polymorphisms in the normal population are very evenly distributed. So they follow a Hardy-Weinberg distribution with about 25% of the population being I homozygotes, 25% of the population being D homozygotes, and 50% being heterozygotes. If you look at endurance athletes, there is a significant skew to the I allele. So you're much more likely to have be an II homozygote if you're a long-distance swimmer, long-distance cyclist, long-distance runner. On the other side, if you look at sprint athletes or weightlifters, there seems to be a skew to the D allele. So if you look at endurance athletes who perform in a hypoxic environment, and in this case on the left-hand side graphic, this is a study of British mountaineers who've repeatedly ascended over 7,000 metres without any medical problems, you can see a profound and statistically significant skew to the I allele. So there are very few individuals falling into this category who were DD homozygotes. If, on the other hand, you look at critically ill patients who are profoundly hypoxemic, and in this case patients with the acute respiratory distress syndrome, and this is Richard Marshall's data from one of our home institutions, University College London Hospital, at the end of the 1990s, there is a relationship between the alleles of the ACE gene and survival. So those patients who are II homozygotes have a mortality of only around 10%, whereas the DD homozygotes have a mortality of in excess of 50%. So a gene which is both conferring a survival advantage in critically ill patients who are hypoxemic is also associated with a performance advantage of climbers at altitude. The second reason that we thought that there was value in doing this type of research at extreme altitude was our concerns about some of the explanations of how people and why people adapt to altitude, so the acclimatization story. And the reason that we were concerned about this story is that it fails to explain the differences that you've seen between individuals as they ascend to altitude, and those differences are large. So there are some individuals, we've already mentioned Reinhold Messner and Peter Habeler, who can successfully summit Everest without supplemental oxygen. There are others who will struggle to get to Everest Base Camp. And so you'd expect any model of acclimatization to explain these differences between individuals. What I'm showing you here is the relationship between altitude and levels of oxygen. So on the horizontal axis you have barometric pressure in kilopascals. On the vertical axis we have altitude in meters, and on the top horizontal axis you've got the partial pressure of oxygen. And as you all know, as you ascend through the atmosphere, the barometric pressure falls. The proportion of that pressure that is oxygen remains the same at around 21%, and as a consequence the partial pressure of oxygen falls, so that by the time you reach the altitude of Everest Base Camp, so around 5,300 meters, the partial pressure of oxygen is down to about half what it would be at sea level, and by the time you reach the summit of Everest it's down to about a third. So 1/2 at base camp, 1/3 at the summit of Everest. Now if you acutely expose people to these kind of levels of hypoxia, so for example the summit of Everest, it's extremely bad for them. And this graph shows the so-called time to loss of consciousness curve. On the horizontal axis you can see altitude in meters again, and on the vertical axis time in minutes. And the red curve describes the time that individuals will remain conscious if they are suddenly exposed to a pressure equivalent to that sort of altitude. And this is research from military aviators. So the original research done around the time of the Second World War. And what you can see is that when you get to the altitudes around the summit of Everest, so close to 9,000 meters, individuals will pass out, lose consciousness in somewhere between 1 and 2 minutes. And very rapidly thereafter they will die. Now intriguingly, at much lower altitudes there are measurable effects of acute hypoxia, and even down to around 1,800 meters, so not that far, not that much higher than the altitude at which you're sitting now at Kathmandu, there are measurable changes in night vision, for example. Now, as we know, individuals can successfully reach very high altitude, and they can do so without supplemental oxygen, and so there are a set of processes that they go through to be able to do that, and those processes are known as acclimatization. And the graphic in front of you, which was taken from a review article in the British Medical Journal towards the end of the 1990s, summarises those physiological processes that are thought to be central to acclimatization. And I've highlighted some of them for you, and they're all about increasing oxygen flux from the atmosphere to the cells in a situation where you're breathing a lower level of oxygen. So if you like, compressing the steps of the oxygen cascade because you're starting from a lower level of inspired oxygen. And it is true that as all individuals ascend to altitude, they tend to undergo these changes. So they increase their heart rate, for example, and that will increase their cardiac output. They increase their ventilation, and that will increase the amount of oxygen that reaches their alveolus. It will blow off carbon dioxide, as a consequence increase the alveolar oxygen tension by the alveolar gas equation, and therefore the driving pressure of oxygen across the alveolar capillary membrane. They increase their hemoglobin concentration, and that will increase the carriage of oxygen within the blood. They also increase their capillary density, and that can reduce the diffusion distance from any particular capillary to a metabolically active cell. Now, the problem with this explanation is that were it a full explanation, you would expect that changes in these individual variables would explain an individual's relative performance at altitude, and that's simply not true. So the individuals who perform best at altitude do not have, for example, the biggest increase in hemoglobin, they don't have the briskest increase in ventilation, they don't necessarily have the greatest increase in their capillaries in their muscles. And probably most surprising and counterintuitive is that the fittest individuals, so those individuals who have the greatest endurance capacity at sea level and therefore are very effective physiological machines in terms of shifting oxygen around the body and down to the cells over a sustained period of time, do not perform best at altitude. And we'll show you data later in the day that clearly supports that. Probably the shining example of this are Reinhold Messner and Peter Haberer, and they were very extensively studied in the 1980s and 1990s, in part to try and explain how they had achieved this extraordinary feat of being the first to successfully ascend Everest without supplemental oxygen. And I won't take you through the data in detail, but probably the easiest way to summarise it is that the most remarkable thing about them was how unremarkable they were. So whether one looked at their overall fitness levels, their ability to diffuse oxygen in the lungs, their ventilatory response to hypoxia or their muscle physiology, they were remarkably similar to controls. And in many of the studies, and you can see a graphic here of 3 sets of data, those controls were relatively sedentary Swiss couch potatoes, for want of a better term. Furthermore, and probably even more surprising to some, is that the oxygen content in the blood in individuals who have had sufficient time to adapt to high altitude is actually maintained or indeed above sea level values. So this is data from our study in 2007 from a paper in the New England Journal, and it's a slightly complicated graphic, but if you focus on the darkest bars, which are oxygen content in mils per litre, you can see that if I take you across the graph from sea level through Everest Base Camp at 5,300 metres, 6,400 metres, Camp 2, 7,100 metres, Camp 3, and then the highest point at which we made measurements, which was 8,400 metres, you can see that oxygen content is maintained at least at sea level values right up to 7,100 metres. And in fact, at Everest Base Camp and 6,400 metres, it is in excess of sea level values. So the fall off in performance, which you very clearly see, and we'll show you the exercise data that backs that up later in the day, is not explained by changes in oxygen content. There is in fact an increase. The elevation of hemoglobin that occurs with acclimatization more than counteracts the fall in oxygen saturation, such that the total oxygen content is maintained or increased. So our core hypothesis is very much explained by these observations, and that hypothesis is that the functional changes that you see in individuals adapting, acclimatizing to high altitude will not be explained by changes in oxygen content. And when we talk about functional changes, we mean both whole body changes, such as VO2 peak, the maximum amount of oxygen that can be consumed at peak exercise, or organ-specific changes. And so we wanted to look at alternative explanations of why individuals, for the differences between individuals, and those included changes in the microcirculation. So in a context where the larger blood vessels have a normal supply, indeed elevated supply of oxygen going through them, there may be problems in the distribution of that oxygen through the smaller blood vessels. Our primary hypothesis was that there would be a change in the metabolic efficiency of the cells, so that the amount of work, and by that from a biochemical perspective we mean ATP, that was generated by any given volume of oxygen could actually vary. And there are good reasons from the basic science literature to expect that that might be possible. And we also wanted to explore alternative cellular and indeed mitochondrial mechanisms. So I'm going to go away from the science for a little while and tell you a little bit about the practicalities of trying to conduct this type of research at altitude. The Caudwell Xtreme Everest expedition in 2007, although it was very prominent and received much media attention, was really only the centrepiece of a much larger programme of research. The picture you can see in front of you is of Cho Oyu, situated on the northern border of Nepal, the 6th highest mountain in the world, just over 8,200 metres. And it's there because we went to Choi Yu twice in preparation for Everest, and that was part of a broader series of expeditions to altitude, so both in the Himalayas and in the European Alps, and studies in environmental and hypobaric low pressure chambers in order to make sure that our team and our equipment and our protocols would work at altitude, and to give us the highest degree of confidence that we could have that when we went to Everest we would be able to do what we'd set out to do. And this programme of study was really all about feasibility and reliability and validity, so being confident that we could make the measurements accurately that we needed to make, and only in small part about obtaining some of the pilot data that we wanted in preparation for Everest. To give you a flavour of the central Caudwell Xtreme Everest expedition, here are some of the stats associated with it. We were lucky enough to have 2 film crews. The first crew produced 2 Horizon documentaries which were shown on the BBC in late 2007. We also had an IMAX crew with us and were awaiting that film in the next year or so. We had 7 laboratories, we had 26 climbing Sherpas, a core team of more than 50 individuals, more than 204 trekkers, 450 of these 60-litre blue plastic barrels that you'll be very familiar with. And all that adding up to around £2.5 million in cost and 26.5 metric tonnes of equipment being shipped out to Nepal. And here are some images of some of the preparatory work we did. You can see the back of a meat van. Although we initially explored using very high-tech environmental chambers, in fact we were able to borrow a meat van from one of our local butchers in return for some sponsorship, we were able to borrow a meat van from one of our local butchers, and that has allowed us to evaluate our equipment down to minus 25 degrees in the car park outside a laboratory in North London. The images on the left-hand side that you can see are of an extraordinary facility in rural England. The metal tube you can see in the lower half of the picture on the left is about 60 feet long and about 12 feet in diameter, and it was designed to evaluate ballooning equipment, so the type of equipment that people like Richard Branson use when they're trying to do a ballooning expedition around the world. That tube can be decompressed to about 100,000 feet, so 30,000 odd metres, much higher than the summit of Everest. And we were able to use it at much lower altitudes, so not somewhat higher pressures. And you can see in the upper picture something called a metabolic calibrator, which is a device to simulate human respiration that we could use to calibrate and validate our oxygen consumption measurements. And you can see probably just make out someone in the background on an oxygen mask doing those experiments. The picture you can see here is of a courtyard about 50 metres, if that, from where you're currently seated. And these are some of the 450 plastic barrels, the more than 100 cylinders of medical gas, and the various other equipment that we shipped out. We had a team of 5 entirely focused on the logistics of this expedition. They had this enormous Excel spreadsheet with more than 500,000 items on it, and they did an extraordinary job. They successfully got every single piece of equipment to where we wanted it when we needed it, and we sometimes somewhat ruefully reflect on that when in our normal clinical practice we're trying to get hold of some device or drug and it takes rather a long time to arrive. We became involved in areas of activity that doctors are not commonly involved with, so this is some of the project management that we were drawn into. This is a spreadsheet describing the location of each individual who came out on our core team, along with in yellow all the individual volunteer treks, and in blue the various groups as they were spread out over the mountain. We also got involved in fundraising and marketing and PR in a way that we hadn't previously been as doctors. And this is the brochure that we produced in 2006. You can see at this time we hadn't met John Caudwell, so we're simply Xtreme Everest. And we got involved in a lot of media contacts. And that was necessary in order to raise the funds for the expedition. It was difficult, to say the least, to obtain funds from the classical medical research funding bodies, but we were very well supported by our partners in biomedical companies. And you can see here a list of those companies that did support us, and I emphasise that all these grants were unrestricted, so we had complete freedom in the science that we wanted to do. The individual you can see in the picture is John Caudwell, who very kindly was the main sponsor of the event, and also, as you can see from his blue outfit, one of our volunteer trekkers, so he was also a subject. Many of you will be familiar with the approaches to Everest, but I'll just remind you of the route from Kathmandu through the domestic terminal at Kathmandu Airport in these tiny little prop-driven aeroplanes up to the airstrip at Lukla, and then everything obviously from there carried by foot or by yak. We ascended up the Dude Cozy to Everest Base Camp, and you can see here the blue dots represent places that where our volunteers and investigators stayed overnight, and the red dots represent places where we had a fixed laboratory. So fixed laboratories at Namche Bazaar, Ferrochet and Everest Base Camp. Here's an image of some of our equipment being taken in the lower reaches, lovely green valleys of the lower parts of the mountains of Solo Khumbu, and then higher up, now getting close to Everest Base Camp in the much more barren regions adjacent to the Khumbu Glacier. Here is Everest Base Camp, and in the foreground you can see the two research tents that we used, in the background the logistics tent, and behind that the 900 vertical metres of the icefall itself. And what we could do at Base Camp was essentially the same as what we were able to do in our research laboratories in London. We had 240 volt AC electricity, and we had all sorts of devices that allowed us to measure things in the same way as we could in a conventional physiological laboratory. So we were able to use bicycle ergometry, breath-by-breath expired gas analysis, cardiac output measurement. We had a retinal camera there. We were able to look at saccadic movements. We were able to make quite complex neuropsychometric measurements. And here's an example of some of those. You can see transcranial Doppler in the upper left image, and in the upper right image pupillometry measuring the reaction time of the pupil to a sudden flash of light. Our volunteers, who were not part of the investigator team, we were relatively kind to, and the most extreme thing they probably did was getting onto the exercise bikes. Our investigator team kindly involved themselves in some more invasive studies. So here are some examples of studies where we used, for example, nasogastric tubes or intra-arterial lines, and unfortunately, in some studies, we needed 2 intra-arterial lines, because when you take samples, you interrupt the measurement signal, so you need one line for sampling and 1 line for measurement. And this is one of my favourite pictures from the expedition. It's not so aesthetically pleasing as many of the mountain pictures, but it is a picture of my wife, Denny Levitt, taking a muscle biopsy from Chris Imray, who I think is with you today. And the great pleasure of this picture is that my wife is an anaesthetist, an intensive care doctor, and Chris is a surgeon. And it's not often you get images like this. So if I now start to take you back to the science, there were 2 subject groups involved in the Caudwell Xtreme Everest project. The first group were the healthy volunteer trekkers drawn from the general population who ascended to Everest Base Camp over 12 days, having flown out from the UK. And were measured initially at sea level and then at various stages on the way to Everest Base Camp. The second group were the members of the investigator team who also went up to Everest Base Camp. And there were 24 of those, and they were studied on a slightly slower ascent, and this is an important distinction. The investigators took 15 days to get to Everest Base Camp, and therefore we never lump the data together. We do on occasions compare these 2 groups, but because the ascent rates were different, we don't pool the data together, and that emphasises a key feature of the design of the study. The ascent profiles of the trekker group were very tightly matched. So they ascended at the same rate, they stayed at the same places overnight, and they kindly agreed to not, on rest days for example, go up and down by more than 300 metres, and as a consequence the differences that we see between individuals, and this is an important point, the differences we see between individuals should relate to their underlying physiology and not to the rate at which they ascended up to Everest Base Camp. The investigator team at Base Camp is further divided into 2 groups. There are the Base Camp staff who stayed at Base Camp for nearly 3 months and therefore give us a window on sustained exposure to hypoxia at this level. And then there's a slightly larger group of 14 individuals who ascended higher on Everest, in some cases to the summit. This is a graphical representation of the ascent profiles, and you can see emphasised again the fact that the trekkers were ascending slightly faster than the investigators, and as you see the data later in the day, you'll see this resulted in important differences in physiology. You can also see here highlighted the period of time, nearly 10 days at Camp 2, where we got much of the extreme altitude data, and also highlighted the fact that we used supplemental oxygen above about 7,000 metres. And that was very simply because we had a clear set of priorities. Safety was the highest priority above science, and then the third was summit or the climbing, and the climbing really only in so much as it allowed us to meet our scientific goals. And as we know that the death rate on Everest is, or the death rate on 8,000 metre peaks is double in those who don't use supplemental oxygen, as a safety measure we insisted that everybody use supplemental oxygen above about 7,000 metres. But importantly, when we made measurements, we made sure that that the subjects had been off supplemental oxygen for at least 20 minutes. To close, I'm just going to, if you like, forestall a couple of frequently asked questions, and they relate to key confounding variables in this study. And we are commonly asked about temperature and dehydration, which are very reasonable questions in relation to individuals ascending to high altitude. This is data from our methodology paper, published a couple of years ago now, highlighting that the laboratory temperatures were really remarkably consistent and not extreme values. And that's because we had very high quality shelters, often two-skinned tents that provided a lot of insulation, and because we did most of the studies during the warmer part of the day. And so you can see that there isn't a dramatic variation between different laboratories. The second concern is that individuals might be very dehydrated, but because of the many plasma measures that we have, I can reassure you that that wasn't the case. We know that subjectively because individuals had free access to water and they subjectively reported that their urine was clear, but we can also know that objectively from the fact that the osmolality is remarkably consistent across the study. Above Everest Base Camp, we had two further fixed laboratories in the Western Coombe at 6,400 metres and on the South Col at just under 8,000 metres, and we made the highest physiological measurements at 8,400 metres, and you'll hear about those towards the end of the day. In order to get to those laboratories, we had to traverse one of the two principal areas of risk. So the two principal areas of risk were the icefall and the summit ridge, and in order to manage the risk in the icefall, we ascended through it as few times as possible and as rapidly as possible and then as early in the day as we could. In order to manage the risk on the summit ridge, we very deliberately climbed to the summit very late in the season, so we were able to have a day in which we weren't exposed to lots of congestion from other parties, but we had the ridge entirely to ourselves. This is the laboratory at Camp 2, and you can see directly behind the Lhotse face and the summit of Lhotse, and to the left the southwest face of Everest. And in this laboratory at Camp 2 we're still on 240 volt AC electricity. We're still able to measure all the things that we would normally be able to do at sea level. This is the Lhotse face. You can see just left of centre a long smooth area we climbed up the right hand side of that. Camp 3 is about 2/3 of the way up that, and then we traverse up over the so-called yellow band to the left up to the South Col. We're on fixed ropes. It's relatively steep climbing at this point, and ultimately we get to the South Col, where again we have a fixed laboratory, this time down to ultra lightweight car batteries. So we're on 24 volts DC, but still with all the devices working because we had gone to the trouble of evaluating them beforehand and validating them in this type of environment. So we knew that they would work, and they would work effectively in both the cold and at the extreme altitude. So I'm going to close there and leave the others to present you some of our results. But in closing, I need to make some very big thank yous. And those are in particular to our Sherpa colleagues, Sherpa friends and colleagues, without whom we simply wouldn't have been able to do this expedition. We would not have been able to get the kit and probably ourselves up to the South Col and beyond. And in addition to the Sherpas, all our other Nepalese colleagues and friends, and in particular our colleagues and friends at the Summit Hotel, who made the expedition possible. I also need to thank these individuals who are trekker groups, more than 200 of them who ascended to Everest Base Camp and kindly gave of their time and repeatedly accepted us making sometimes quite complex measurements, and again, without them we couldn't have done the study. And the last group, of course, are the investigators, and here are some of the investigators at the Summit Hotel just before we headed off up to Everest Base Camp in the spring of 2007. So I thank you all for listening. Once again, I regret not being able to be with you, and I really hope you enjoy the rest of the day. Thank you.